Dear friends, today we are going to discuss some fundamental aspects regarding evolution and stratigraphy. To what extent does this special perspective of geological time gives us an insight into the nature of evolutionary processes? Most particularly through paleontology, it is possible to trace phylogenies, that is the descent of particular evolving lineages. This has in the past been one of the major tasks for the paleontology, and nowadays not only phyletic evolution but also community evolution is receiving much attention. On a more general scale, a unique contribution of paleontology is an understanding of how evolutionary rates have varied over extended time periods, and from this has emerged a conception of modes of patterns of evolution. A further possibility is that the predictions of genetical theory can be tested, as already been explained with the allopatric evolution. The effects of both inter and intraspecific competition can be seen in fossil as well as in modern taxa. Clearly, the better the fossil record is known in terms of the distribution of the past invertebrates, the greater the possibilities for this kind of work. Some of these topics are here selected for special discussion. Intimately connected with rates of evolution are the various patterns in which animals arise and evolve. It is here that fossil record is of particular value. For the nature of these patterns might be predicted, it would be unlikely to be confirmed from observations in modern animals alone. Two main grades or modes of evolution have already been referred to in passing and will be summarized here. These are micro and macro evolution. The origins of new species from existing ones, largely through changing interactions between the animal and its environment, are generally referred to as the microevolution, although the genetic and isolating processes by which species differentiate are well understood. It is only comparatively recently that full confirmation of how they have actually operated through time has been clearly seen. Simpson in 1944 and 1953, working mainly with vertebrates, linked genetic theory and paleontological data in a timely manner, as did Carter in 1954. But for a long time, most paleontologists tended to have firmly entrenched the concept of evolutionary change based on somewhat priority arguments. There was a general tenet that species had arisen by what came to be termed phyletic gradualism. This implies an even slow and methodological change of the whole gene pool and hence of the characters of the entire population. This transformation was thought to have involved most or all of the population and to have occurred over much of the geographical range of the parent species. If this were true, one would expect the fossil record, that is ideally at least, to consist of a continuous series of graded intermediate forms. Now, there are a few instances where this may have happened. For example, the evolution of the Cretaceous sea urchin Micraster may in part be based upon such processes. This is disputed by some. But in fact, such gradual transformations are not commonly encountered in the geological record. Indeed, the situation is quite the reverse. In most continuous rock sequences, what is normally seen is a sequence of stable fossil populations separated by abrupt morphological breaks. It is true that at many sedimentary sequences there are innumerable undetected breaks in this sequence, that is diastems, which represents periods of erosion sandwiched in between depositional interludes. But do diastems or unconformities, other imperfections of the geological record, and the migrations of the faunas into the area represented by the sequence under consideration account for all the apparent imperfections of the fossil record. It would have been so accepted some years ago. Such arguments had become part of the standard habits of thoughts or paradigm of most paleontologists. In 1972, Eldridge and Gould, having independently documented precise patterns of microevolutionary change in Devonian trilobites from eastern North America and in Pleistocene land snails from Bermuda, concluded that old arguments were largely untenable and were in any case rather out of touch with genetic theory. Instead, they made clear that evolution by allopatric speciation was a more viable model for paleontology and could sometimes be tested by a close examination of fossil populations. The central concept of allopatric speciation is that new species can only arise when a small local population, one or more gamedomes, becomes isolated at the margin of geographical range of the present or parent species. Eldridge and Gould termed these small marginal populations, that is, peripheral isolates. It is in these, as we have seen in previous sections, that gene flow is at its most rapid, whether under the influence of selection or of drift. Should the parent species then become extinct, or one of the peripheral isolates may migrate into the original range of the ancestral species and then expand rapidly. In the rock sequence, this replacement would be represented by an abrupt faunal break. This happened more than once with Eldridge's trilobites and also with Gould's Bermudan land snails. Indeed, the relative degree of isolation of the islands the latter lived on at any particular time could be inferred from geological record. Most fossil species tend to exist in punctuated equilibria, that is, as relatively stable populations showing little change during their lifespan and separated from each other by abrupt faunal breaks suggestive of extinction, at least for that area, and migration of the new stock into the region. Although these concepts had a substantial impact on paleontological circles after Eldridge and Gould's paper, they were not in themselves really new. They may in fact be traced back to some tentative and largely ignored comments by Darwin. In their present form, the ideas are owed to Mayer, who in 1954 wrote that rapidly evolving peripherally isolated populations may be the place of origin of many evolutionary novelties. Their isolation and relatively small size may explain phenomena of rapid evolution and lack of documentation in the fossil record, hitherto puzzling into the paleontologist. This lack of documentation has always been a problem for only in relatively rare examples where the successive populations are widespread and preserved through great stratigraphical thicknesses. Can allopatric speciation directly be seen to have operated? The existence of punctuated equilibria by itself is consistent with almost any model of speciation, and very often little can be inferred from a particular vertical sequence as to how a species actively arose. Even so, allopatric speciation seems to be a more valid model for paleontological evolution than the old concept of phyletic gradualism or stately unfolding as it came to be known, and as such it has found great favor in recent years. Though it is not without its critiques, because a fair degree of caution is needed in extrapolating evolutionary events directly from the rock record. For since the early days of the punctuated equilibrium versus phyletic evolution models, it has become increasingly clear that paleobiological events linked as they are two processes of sedimentary deposition are often obscured by the rock record itself. In other words, our knowledge of small-scale evolutionary events is only as good as the sedimentary record allows it to be. There is an ultimate limit to paleontological resolution in most depositional settings because sedimentary processes are either too slow or the final record too intermittent to allow preservation of critical biological events. It has came by the work of Sneedle 1980 and 1982. Johnson in 1982 again makes the point that since evolution has been mainly a process of adaptation to changing environments, the apparent phyletic or punctuational evolutionary changes seen in the rock record may in fact reflect nothing more than relative rate of environmental change. Punctuational evolution is more likely to seem to have taken place in benthic animals whose environments were subject to abrupt changes. Gradual evolution, on the other hand, is more evident in pelagic organisms inhabiting environments which were far less prone to such sudden fluctuations. In short, the punctuational versus phyletic models is not just a simple division into two different kinds of event. It has been said, and to good effect, that one man's punctuated equilibrium is another man's phyletic gradualism, and this of course depends on the scale at which the events are studied. What will be essential in future studies is new data derived from detailed microstratigraphic sampling, in other words by bedding plane collection through continuous sequences. And whereas, as Sneedle 1980 has put it, even the most carefully sampled microstratigraphic sequences represent spans of times longer than human lifetimes. This kind of detailed work gives a potential source of data for paleontology and evolutionary biology which could not be gained by studying living populations. And it is in this field that much future research will surely lie. In spite of these cautionary comments, the model of evolution by allopatric speciation still holds good and makes more sense than the splitting of lineages by phyletic gradualism. Phyletic changes have been more important in the fine-tuning of new species precisely to their habitats subsequent to the origin of ancestral group allopatrically. Size increases through time, for example, well documented in many vertebrate and invertebrate lineages, may be interpreted very largely in terms of the phyletic model. The origin of species is thus seen as a case of quantum evolution at the species level. The isolated population has to pass out the set of ecologically precise parameters to which it and the parent population are adapted into another adaptive zone. The intervening conditions are unstable and inadaptive. Most, and indeed usually all, populations undergoing such a transition will not survive to gain the next adaptive peak. It is convenient to consider the problem in terms of an adaptive landscape that is right in 1932 of high hills of adaptive fitness separated by low-lying inadaptive swamps which have been traversed and in which most adventurers will sink without trace. Those that do survive and gain the next adaptive peak will carry only a fraction of the total gene pool of the parent population. Thus, this new isolated population already starts with a biased sample of genes by comparison with whole gene pool, Mayer's founder principle, and this in itself subsequently reinforced by mutant alleles is a significant step towards speciation. Coming to the third module, that is macroevolution. Evolution is an hierarchical phenomenon in the sense that changes of evolutionary importance may take place at any level. The genome, the individual, the species, or above. Moreover, processes such as selection and drift can operate at all these levels. We have already considered some of the mechanisms operating at the genome level, as we have seen that microevolution is concerned with changes within species or with minor taxonomic transitions. Macroevolution, on the other hand, refers to the evolution at higher levels, or to use that is Levinton's 1983 definition, with the characteristic the transitions that diagnose differences of the major taxonomic rank. It is in the study of macroevolution that paleontology has perhaps its most direct contribution to make, for bearing in mind that several thousand generations may have lived and died during the time of deposition of only a millimeter or two of the sediment. There is no way in which a truly fine focus can be obtained on events which took place during this time unless sedimentation was very rapid. Detailed paleontological studies are better at resolving more major features of evolution. Species selection, the origin of new structural plants and higher taxa, adaptive radiations and extinctions, and rates of evolution. Coming to the species selection, we begin with the species selection since it is here that long-term differences between micro and macroevolution are most clear. In microevolution, the unit of selection is the individual, of course, or individual variations arise from mutations and recombination, and natural selection operates by favoring individuals with character combinations suitable for survival. At the macroevolutionary level, however, as Stanley 1979 has shown, the unit of selection is the species. The source of variation is in rapid speciation events, and selection acts upon species with favorable character combinations. This is species selection. It is of course analogous to natural selection, but while the rate of reproduction and potential for individual survival operate at the individual level, rates of speciation, that is birth of new species, and species longevity are determinants in species selection. So having defined species selection, how does it operate? On the punctuation model, speciation events produce discrete species which after a brief and geologically instantaneous period of flux have their characters defined from the start. Thereafter, there is usually a long period of little change. Virtual stasis, except perhaps for a little phyletic fine-tuning within a plexus of related species, however, when followed through long time intervals, this is often a net evolutionary change or trend. There may be several coupled trends, some of which can be reversed. Such trends, for example, the increase in complexity of the ammonite sutures or the numerous changes evident in graptolite phylogeny, have been known for a long time and have been interpreted in various ways. In modern microevolutionary theory, these trends result from species selection. Those species with character combinations advantageous for survival being selected through time. How rapidly and how effectively they spread depends upon many factors, not least of which is the rate of generation of new species which happen to have these characters and the tendency to survive for long periods. Variations in speciation rate and in extinction rate alone, or with selection operating, will affect the development of a new evolutionary plexus or clade. An important concept stressed by Stanley is that microevolution is decoupled from microevolution. This is because individuals are the units of selection at the microevolutionary levels, and the species are the equivalent selection units at higher levels. Hence, large-scale phylogenetic trends are nothing to do with the phyletic trends within a particular species. Through species selection is the basis of much macroevolutionary or microevolutionary change. It is not the only agent. There is also what are known as the direct speciation, where basically random changes move in the same general direction, and phylogenetic drift, which is comparable to genetic drift at a macroevolutionary level. These are less important, however, and species selection remains as macroevolution in action, the primary mode of macroevolutionary change over longer time periods. Coming to the origin of higher taxa. New types of structure and new grades and systems of animal organization appear very suddenly in the fossil record. testifying to an initial and very rapid period of evolution. Such changes may be relatively small, defining new groups at lower taxonomic level. Much larger changes of great importance result in the origination of higher taxa, orders, classes, and phyla. Even in the first representatives of these groups, all the systems of the body and all the anatomical, mechanical, physiological, and biochemical elements therein have to be precisely and harmoniously coordinated from the very beginning. The origination of higher taxa in this sense remains at least the least understood of paleontological phenomena. It is sometimes referred to as the mega-evolution, though the term is seldom used nowadays, as it seems to have been another form of macroevolution operating at a higher level. And it is hard to know where to draw the boundary. When more is known about the functions of the genome and how the information carried in the genetic code results in a fully developed individual, we shall have more of an insight into this most critical yet most elusive of all aspects of evolution. The links between the higher taxa are obscure and are but poorly represented in the fossil record, as exemplified by the diversification of life in the early Cambrian, where transitional or linking forms are absent. The geological record gives no indication of such relationship, and our knowledge thereof rests entirely upon the traditional disciplines of comparative anatomy and embryology, supplemented to some extent by chemical taxonomy. But what the fossil record does give is many examples of the instantaneous origin of new structural plans. The derivation of monograptids from diplograptids, the beginnings of the irregularity in the echinoids by the abrupt displacement of the periproct, the fusion of the mantle in bivalves to form siphons, all these are important. Sudden key innovations which allowed great evolutionary diversification thereafter. Such changes as these are now understood in terms of changes of regulatory genes which control the operation of structural genes. Most importantly, evolution or evolutionary novelties may arise by relatively minor changes in the regulatory gene complex. This might affect a character or a group of characters, but such regulatory gene changes might, on the other hand, affect the whole organism. A well-known process that is presumably controlled by regulatory genes and seemed to have been important in evolution is a change in relative growth of different parts of the body, giving the derivative stock different proportions. Such allometric changes might be sudden or gradual and might well account for much evolutionary change. Abrupt and major changes are less easy to understand and have been the subject of much discussion. Many years ago, Goldsmith proposed a novel and certainly somewhat fanciful concept to account for the seemingly instantaneous origin of new species and indeed higher taxa. He held that major chromosomal mutations or rearrangements could in rare cases produce a hopeful monster, a viable new kind of organism which could be the progenitor of an entirely new taxon with its own character. Goldsmith's views were never popular, largely because of the very low probability of such a freak, even if it happened to have new adoptive characters being able to find a mate and breed. Yet there is still some support for a modified and toned down version of the hopeful monster concept, and it is not impossible to envisage a viable and not too freakish freak breeding initially with similar novel offspring of the same parent in a small population as the founder of the new taxon. Few would accept Goldsmith's idea in its original and extreme form, but it is quite possible that a comparatively few quantum events or several generations could have led to an entirely new type of organization. Moreover, the modern view of the genome as a hierarchically organized and dynamically interactive system allows for much more coordinated flexibility than would have seemed possible a few years ago. Some kinds of abrupt changes can be more easily understood in terms of changes in the relative rate of development of functional structures. This is known as heterochrony. The most important concept here is the pedomorphosis, the relationship of ancestral juvenile characters into a descendant. This can, according to Gold 1977, take place in two ways. In neoteny, the speed of morphological development throughout ontogeny slows down if then sexual maturity is attained in the descendant. After the same length of time has elapsed as in the ancestor, the stage in development will not have gone so far. In this way, the adult of the descendant resembles the juvenile of the ancestor naturally. Coming to the progenesis, also a progenesis retaining ancestral juvenile characters refers to the precocious onset of sexual maturity. This means that development ceases early and usually the progenetic form is smaller than its ancestor. In neotenic species, however, the adult may be the same size or even larger than the ancestral form. While progenesis affects the whole organism, neoteny does not have to do so. Particular characters or combinations of characters may by themselves be affected because their rate of development is modified so that the full ontogeny thereof is not completed. This process can substantially change the appearance of a whole organ or complex of characters and seems to be very important in evolution, much more so than has generally been appreciated. Neoteny can account for a number of sudden and abrupt changes which could not be fully understood otherwise, including structural and functional changes in whole organisms. Progenesis has been invoked with great success in understanding the origin of early fish-like ancestors of vertebrates from the tadpole-like free-swimming larvae of sea squids. Likewise, the small planktonic copepods may be progenetic products of the easily or early onset of maturity in planktonic larvae of benthic crustaceans. Numerous examples of progenesis in trilobites are known, especially from the Cambrian, and in the geological record, pedomorphosis may well have been very important to all evolutionary grades. Coming to the pedomorphosis, pedomorphosis may be seen as a special case of pre-adaptation. The concept of pre-adaptation in its simplest form means only that animals must already possess characters capable of being modified if they are going to be able to adapt to new environments. Sometimes these characters may be in the form of organs that have lost their original functions. They are therefore spare and can readily become adapted without inhibiting an existing function. More often, however, they are already functional, but by becoming modified, they may then become more flexible in the variety of functions they perform. There are many good examples drawn from the vertebrates, such as the use of the gut diverticulum as an air-breathing lung, Devonian, mid-Devonian, and lungfish living in rivers and ponds that are liable to dry up. The same structure became swim bladder in ray fin fish. Numerous examples could be cited in the invertebrates also. The remarkable adaptation of already existing spines, cilia, and tube feet in echinoids for deep burrowing being an ideal case. However, not only may structures be pre-adapted before modification, they must also become post-adapted while their function and probably form as well as are in the process of being transformed. Pre-adaptation has become recognized as a very critical concept. Indeed, Carter 1954 has written, "No adaptation is possible without some grade of pre-adaptation in the structure to be adapted." An earlier concept relating to the ontogeny as a tool for understanding evolutionary relationship was that of the Haeckel, who believed that the ontogeny of any animal was a more or less direct recapitulation of its phylogeny. In other words, the young stages of the development of any animal are very similar to what the ancestors of that species were like. In embryology and in paleontology, there is a very little evidence for this, and since the widespread acceptance of pedomorphosis, Haeckel's biogenic law, as it was called, has been very largely discarded. Many species are co-adapted to life in an intimate association with another species, usually of a totally different kind. Such associations have arisen by a kind of complementary process known as coevolution, and the adaptations of the two species concerned are essentially linked. A predator-prey system is one such example, as are symbiotic and commensal relationships. Perhaps the clearest of all, the mutually necessary relationships between flowering plants and insects, or in some cases birds. Whether such co-adaptations were achieved gradually or in quantum steps is a matter of continuing debate. We have discussed that the evolution is of two kinds, microevolution and macroevolution, and of two types also, or two modes of evolutions are there. One is punctuated equilibrium and another is phyletic gradualism. Actually, the evolution is a slow and gradual process when the changes tends to accumulate in an organism, and then suddenly the things starts moving differently, fastly, and on a different plane. Then you have a new species being arise from the previous one, and then that process what you call as evolution. So it is a time dependent process and is of many scales which can operate at the species level or at the level of the higher taxon. At the smaller level it is known as microevolution, while at the higher level you call it as a macroevolution. Thank you very much.